The growth cone of an elongating axon navigates the axon's pathway to its synaptic target. Surface receptors on filopodia and lamellipodia detect extracellular molecules that guide axons towards their targets. Growth cones turn towards attractive guidance cues with a burst of actin filament polymerization that pushes the growth cone's leading margin towards the attractant. The attractive cue nerve growth factor, NGF, activates regulatory proteins such as ADF and cofilin, which create new sites for actin polymerization, and the ERM protein rhodixin, which links new actin filaments to the plasma membrane at the growth cone leading edge. In an NGF gradient, ADF cofilin and ERM proteins are locally activated to direct the actin polymerization that pushes the growth cone leading margin towards the NGF source. NGF also increases adhesivity of the leading edge, as activated ERM proteins bind and concentrate the adhesion molecule L1 on filopodia. In addition, the extracellular matrix receptor beta-1 integrin concentrates at filopodial tips. The significance of ERM-mediated linkage of actin to the growth cone leading edge is indicated by experiments to inhibit ERM in growth cones. In ERM-inhibited growth cones, actin filaments were reduced and were abnormally distributed in central growth cone regions. In addition, the retrograde movement of actin filaments from the leading edge was accelerated when F-actin links to the plasma membrane were reduced by ERM inhibition. And in response to the addition of NGF, new F-actin barbed ends were mislocalized to the growth cone center. Not surprisingly, growth cones with reduced ERM function did not turn towards an NGF attractant. Thus, ERM proteins like rhodixin mediate growth cone responses to attractive guidance cues by linking new actin filaments to the plasma membrane to efficiently protrude the growth cone leading margin towards the source of an attractant.